Hello everyone. So today we are going to see two important questions. The first one is what is Bohr effect, and the second one is how is carbon dioxide transported in our body. Okay, so what is Bohr effect? Let's see. The Bohr effect is it is the effect of partial pressure of carbon dioxide or H positive ions on the oxygen dissociation curve. Okay, now this is the oxygen dissociation curve. Okay, so this is the oxygen dissociation curve, and we have seen earlier that the oxygen dissociation curve or ODC curve is a sigmoid shaped curve. Okay, and why is that? Why is it a sigmoid shaped curve? Because binding of one oxygen to the heme group facilitates the binding of other oxygen molecules to respective heme groups. Okay, so that is the reason why the ODC curve is a sigmoid shaped curve. Now, we have seen that with increase in partial pressure of carbon dioxide or H positive ions or a decrease in pH, this results in shifting of ODC curve to the right. Okay. So if the, let's say the starting pH is 7.6. Now as the pH decreases, okay, or H positive ions increase, the oxygen dissociation curve is being shifted towards the right side. Okay, so 7.6 it is here, 7.4 it shifts towards the right side, and 7.2 it shifts further towards the right side. Now what happens when this shifting takes place to the right side? Okay, this results in dissociation of oxygen from hemoglobin. That means the bond between the hemoglobin and oxygen is loosening up. Okay, more and more of oxygen is given out from the hemoglobin, or dissociation of oxygen from the hemoglobin takes place. Okay, for example, let's say this is the partial pressure of at tissue level. Okay, 40 mm of Hg. Now if you see over here, when the pH was 7.6, okay, so you can see over here approximately 80% of hemoglobin is saturated with, okay, it is over here approximately, yeah, so approximately 80% of hemoglobin is saturated with oxygen. Okay, now when the pH decreases to 7.4, the saturation also decreases. Okay, and roughly around 7.2, the saturation is near about 60. Okay, so when the pH was 7.6, more hemoglobin was saturated with oxygen, and when the pH decreased to 7.2, then less of hemoglobin is saturated with oxygen. As a result, oxygen is lost when the pH decreases, okay, or H positive ions increase, or if the partial pressure of carbon dioxide increase. So this is known as Bohr effect, shifting of oxygen dissociation curve on the right side, which signifies increased unloading of oxygen from the hemoglobin, and this unloaded oxygen is given off to the tissues. Okay, now why does this happen? Okay, this happens is because firstly the hemoglobin, the oxyhemoglobin is more electronegative, so it attracts the H positive ions. Okay, then also when the carbon dioxide which is released from metabolism, it enters the RBC. Okay, it forms carbonic acid, and some amount of carbon dioxide also forms carbamates with hemoglobin. Okay, so carbamates again they stabilize the T form which we had seen in the earlier lecture, T form or a taut form of hemoglobin. Okay, so that decreases the affinity of hemoglobin towards oxygen. Okay. So as the carbon dioxide comes inside the RBC, with the help of enzyme carbonic anhydrase, it forms H2CO3, which dissociates into H positive ions and bicarbonate. So these H positive ions are then buffered by deoxyhemoglobin. Okay, it is buffered by deoxyhemoglobin. Why? Because hemoglobin has a lot of histidine amino acids. Okay, so it buffers the H positive ions, and oxygen is liberated, which is utilized by the cells. Okay, so the bicarbonate which is produced, it comes out of the RBC. Okay, and as a result, to maintain the same electrical neutrality, the carbon chloride ions it goes inside. Okay, so this is known as chloride shift. So this is all about your Bohr's effect. Okay, how pH, decrease in pH or increase in H positive ions or partial pressure of carbon dioxide affects the oxygen dissociation curve. Okay, so if there's an increase in all these, increase in H positive ions or partial pressure of carbon dioxide, the ODC shift towards the right. If it is decreased, in partial pressure of carbon dioxide, or decrease in H positive ions, or increase in pH, okay, then the ODC shifts towards the left. Now let us see our next question. How carbon dioxide is transported in blood? Okay, so basically it is transported in three forms. First is the dissolved form, the second is the isohydric form, and the third one is carbaminohemoglobin form. Okay, so the dissolved form is seen, it's approximately 10% of carbon dioxide is transported in this form. The majority of carbon dioxide is transported in isohydric form, and 15% is transported in carbaminohemoglobin form. Okay, that we will see. Now the dissolved form of carbon dioxide, okay, it is seen in the blood. Okay, so the carbon dioxide is roughly 20 times more soluble as compared to the oxygen. Okay, so the carbon dioxide is just dissolved in blood, and that is how it is transported at the same partial pressure as that of the oxygen. Okay, now when this carbon dioxide goes inside the RBC, then the carbon dioxide, it combines with water. Why? Because there is an enzyme in the RBC called carbonic anhydrase. So this carbonic anhydrase then takes the carbon dioxide, combines it with water, and forms carbonic acid. Now this carbonic acid spontaneously dissociates into H positive ions and bicarbonates. Okay. Now inside the RBC, okay, there is hemoglobin which is already carrying the RBC. Hemoglobin which is already carrying the oxygen. Okay, so this oxyhemoglobin is more electronegative. So as a result, it attracts the H positive ions. Okay, so H positive ions are then attracted towards the oxyhemoglobin. The oxygen is released, and the hemoglobin is buffered by the deoxyhemoglobin. Okay, so this hemoglobin, deoxyhemoglobin, which has again it's buffered by the H positive ions, is buffered by deoxyhemoglobin. Okay, by the histidine amino acids which are present in the hemoglobin. Okay. Why is it called isohydric? Because the H positive ions, even though there is increase in H positive ions, the pH does not change. Okay, that is why it is called isohydric transport, because H positive ions are buffered. Okay. Then the bicarbonate which is formed, it moves out. Okay, and chloride shift occurs, chloride moves in. Okay. In the lung, opposite occurs. Okay. So in the lung, what happens is there is increase in partial pressure of oxygen in the lungs. So the partial pressure of oxygen, it breaks the salt bridges. Okay, as a result, the H positive ions, it is increased. The H positive ions is released from the hemoglobin. Okay, so the H positive ions is released. So this H positive ion again combines with the bicarbonates. Okay, the bicarbonates from extracellular, it goes inside, comes inside the RBC. Okay, and chloride moves out, just the opposite. So the bicarbonate which has come inside the RBC, it combines with the H positive ions to form as carbonic acid. Carbonic acid again dissociates into water and carbon dioxide, and this carbon dioxide is exhaled into the air. Okay, so in the lungs, opposite occur. When the oxygen is inhaled, see this oxygen breaks the salt bridges, hemoglobin binds with the oxygen, and the H positive ions are released, which ultimately forms carbonic acid, and carbon dioxide is exhaled out. Okay. The last one is in the formation of carbaminohemoglobin, which is responsible for about 15% of transport of carbon dioxide. Now this also occurs inside the RBC. So the carbon dioxide it comes inside the RBC, it binds with the terminal amino groups present in the hemoglobin, okay, and form carbamates. Okay, so the hemoglobin binds with, in other words, hemoglobin binds with carbon dioxide and form carbamates. Now these carbamates, they stabilize the T form or the TOT form or the deoxy form of hemoglobin, and this helps further unloading of oxygen to the tissues. Okay, so hope you have understood. Thank you very much.